Okay, I think we're ready to get started, everyone. Welcome. Thanks for being here. Thanks to all of our invited teams, our judges, and to our online community, which I hear there's quite a number of them, getting on towards 100 people. So welcome everybody to ARSX 2024 Safeguarding Sources Challenge. Our theme is the quest to safeguard and sustain genetic diversity. Each year, the ARSX executive team, including the Office of Technology Transfer, Assistant Administrator, Dr. Brian Nakanishi, ARS Deputy Administrators, Dr. Pam Stark-Reed and Dr. Jeff Silverstein, and our ARS Administrator, Dr. Simon Liu, select a topic that resonates deeply with the scientific and agricultural community. This year, we're focusing on a critical issue that could transform agriculture: the sustainable preservation and management of genetic diversity. The theme for this year's challenge invites us to create smarter, more sustainable systems to ensure agricultural resilience and promote innovation within our food system. Today, we will explore solutions to sustain and enhance genetic diversity of plants, animals, and microbes. We put forth this challenge to all of our ARS scientific staff, calling on them to devise innovative and unconventional approaches in one of our pivotal subcategories: advanced collections technology, sophisticated management systems, innovative characterization methods, and revolutionary preservation techniques. Today, we will explore solutions. To explore the proposals you'll hear today have been evaluated by a diverse group of preeminent researchers, science leaders, innovators, and entrepreneurs. This esteemed panel of 5 judges will select the top 3 teams, each receiving up to $100,000 to demonstrate proof of concepts over the next year. So now I'd like to ask our judges to stand. And next slide, please. Please, everyone, welcome Kathy Boomer, Scientific Program Director at the Foundation for Food and Agricultural Research, Dr. Steve Kresovich, the professor at Clemson and Cornell Universities, Renee Vasalos, Director of Agricultural Innovation at the Nature Conservancy, Dr. Steve Kappas, retired Office of National Program Associate Administrator and currently at the Office of Director as a Senior Advisor, and our own Administrator, Dr. Simon Liu. Welcome, everyone. Please give a hand to our judges. So we're just gathering our judges for a picture, so we make sure we get some shots before they have to leave. What do you prefer?
I love the ARS logo.
Do we want to go back a couple of slides for just have the logo behind them? Oh, turn it off. Okay. Thank you. Thanks to all of our judges. So if we can move to the agenda. So we have a packed agenda this afternoon where each of you and those who are joining us online will watch our ARSX 2024 visionaries pitch their transformative ideas. By the end of today, we will know which 3 teams will receive $100,000 to pursue their groundbreaking projects. And now it's my pleasure to introduce Dr. Simon Liu to share some opening remarks for the kickoff of our live event. Thank you.
Thank you very much, Dr. Kim Cook. Welcome. Good afternoon, everybody. Welcome to be in this exciting event here. So ARSX, as you know, that the main purpose of this competition is looking for new idea for high reward, high risk. So everything based on just one word, innovation. As an ARS, as an agency, we know our mission is really to become the leader, the global leader. That's our vision, the global leader in ag discovery through scientific excellence. That is our vision statement. The vision statement is very clear. In order to have a scientific discovery, we need to have innovation. So innovation is the base of everything we do here. So this is the major, I would say, the theme for ARSX. Now, as a research organization, we need to continue to innovate in terms of our approach. In terms of our approach, our method, our technology, and different things we do. The only through innovative way we will be able to continue to improve, not only improve our research, but also to explore the new frontier, to advance our research and to speed up our scientific discovery. That is the whole purpose. Now, ARS, one main job for me as the administrator, I like to make sure that ARS, we have enough or a comprehensive infrastructure. We call it innovative infrastructure, so that we can encourage the innovation. Not only encourage, but also invite innovation. Not only invite, but also to implement the innovation. So as a result, we do have a comprehensive program. I just like to name a couple of them so that you have a chance to look at other opportunity. Of course, ARSX is one of them. Another one is we call it Innovation Fund, which is the fund we receive from our patent, our licensing, and we reinvest those royalty fee into our research program. We call it Innovation Fund. That fund is managed by OTT, the Assistant Administrator, Brian Nakanisi is sitting right here. He's the person managing the Innovation Fund. I encourage all of you here to consider that also. Those fund competition not mutually exclusive. In addition to that, headquarters, we also invest the money into the innovation. That is the headquarter postdoc project. Every year we hire postdoc, but headquarter, we do have a few postdoc that is more prestigious and more consistency in terms of our research theme and also the funding. Every year we sponsor 40 different postdocs. A postdoc is good for 2 years. I believe right now is a postdoc is more than 200,000 overall. And 40 of them you're talking about, we invest millions of dollars to encourage, make sure that we do have innovation platform. So here's my goal. My goal is that for ARS, innovation is a part of our DNA for not only individual as a person, as a researcher, but also for each project. Each project need to have innovation in their mind. How can we continue to break through, to explore the new frontier, find a new method so that we can accelerate our scientific discovery? So today, thank to all the 9 teams. You all done fantastic job. Congratulations to all of you here. To me, even though today we're going to select 3 winners, but each and every team of you are a winner. You are a winner. We have received 25 applications and then go through several different process. We will narrow down to 3 individual applications. But again, make it to this run, you are a winner already. Thank you. Your presentation are so impressive. Personally, I'm so encouraged, so impressed by all of your presentation. And also for the people sitting behind the scene to make this happen. Thank you all. Thank you. That is a job well done. Thank you.
Thank you for those comments, Dr. Liu, to get us started. And now I'd like to invite Dr. Jeff Silverstein up to provide some background about our ARS program. Thank you. ARSX.
Thanks. Thanks. First, I wanted to thank Dr. Liu and the administrator's office for the great support for ARSX for 5 years now, and recognize the executive team I share with Pam Stark-Reed and Brian Nakanishi. But especially, I want to thank all the competitors here. You really make ARSX happen. And so thanks to all of you here and the teams that aren't here today that make this possible. Next slide. So, this is high risk, high reward competition. And I just wanted to share this quote from Wayne Gretzky, you miss 100% of the shots you don't take. So really appreciate the folks that applied and submitted their application. And, for the 5th year, we've had a really excellent response. And I think it's making ARS a little bit more used to the high risk, high reward research. It's a good part of our portfolio. Next slide. So first, let me mention there's the QR code showing up on the screen right now. And for people online or in the audience, this QR code will take you to a page asking for ideas for future ARSX challenges. So we're soliciting your ideas for what would be a good ARSX challenge. And this picture is to remind me to just give a little bit of a overview of the origin story of ARSX. So way back in 2018, I was part of a team that envisioned ARSX. We were thinking about a different platform for innovation other than the Grand Challenge Synergies program that had just kicked off. We were looking for something that was really aiming for breakthrough innovation, innovation that was more like a 10x than 1.5x change. And at that same time that we were talking about this, a AAAS fellow that we had here working with us on innovation at the time, Sarush Parsa, who's standing next to me at ARS at the X Prize competition, he submitted a X Prize proposal for an agricultural X Prize. And Sarush Parsa was selected as a visionary, and he invited me to go with him to Pasadena for the pitch competition. So we got to experience that pitch competition, sharpen the ideas, and really sharpen the whole idea for a X Prize type competition. As a nice complement to this whole experience, Dr. Jacobs Young, who was the ARS administrator at the time, was invited judge by X Prize. So we were all there together, and that's Peter Diamandis, who was leading X Prize at the time. So it got us all talking and thinking about how we could, set up this ARSX competition. We wanted a competition that would invigorate high-risk, high-reward research at ARS. And so we worked with Dr. Jacobs Young and polished up our ideas. And we were really eager to test it out. So now, after 5 years, we know that we have a competition that inspires high-risk, high-reward research ideas. So far, over 5 years, we've had about 170 teams compete for $300,000 prizes each year. And we look forward to growing this competition to keep ARS providing novel solutions for agricultural problems. Some ARSX teams have been very successful in the previous 4 cycles. We've had external grants awarded following up on ARSX teams totaling over $15 million. There have been several patents filed, new research agreements made, external partners found, and new companies founded. So aside from these technology transfer successes, scientists have also gotten recognition from their peers. They've been nominated for Innovative Scientist Awards, and they've been invited for keynote speaking engagements. So ARSX has really become a recognized emblem within ARS as a place to share novel ideas, and it's a great forum for extending our basic science research. So after 5 years, let's see what the next 5 years have to offer. But thanks very much.
Thanks, Dr. Silverstein. Thank you for that background. And now we continuing in the tradition of ARS innovation, it is my pleasure to introduce the first of our ARSX teams. Welcome, Team 1, Bug Banking, Cryopreservation, and Insemination.
Great. Good afternoon. My name is Courtney Grula, and I'm a research entomologist at the USDA in Fargo, North Dakota. Today, I wanted to discuss an exciting area of research for our grant, which is insect cryopreservation and artificial insemination. Next slide. Insects are vital for our ecosystems and food security, yet we're seeing a troubling decline in their populations, biodiversity, and genetic diversity due to habitat loss and climate change. This loss threatens ecological balance, and this is especially concerning because the economic value of insect pollinators is an estimated $170 billion annually. But there is hope through innovative approaches like cryopreservation and artificial insemination. Next slide, please. This technique allows us to preserve insect cells at very low temperatures, essentially putting them on pause for future use. Currently, there's only one insect species in gene banks, that's the honeybee, but we aim to create a comprehensive gene bank for at-risk species, including the monarch butterfly, the critically endangered Sacramento checkerspot butterfly, and several bumblebee species. We are the preeminent lab that can cryopreserve insect species. As you can see here in this slide, this is a successful cryopreserved sperm sample from a monarch. All of these green squiggly lines are monarch sperm cells, and everything fluorescing green is actually alive. So you can see that we had nearly 100% success rate in our cryopreservation technique. With this grant, we will develop artificial insemination protocols using cryopreserved samples. While we have mastered preserving germ cells, our challenge is ensuring these techniques will produce viable offspring. By leveraging the expertise of our team, we can help restore insect populations before they reach critical levels. Next slide, please. Insect cryopreservation and artificial insemination are essential tools in our fight against biodiversity loss, and together we can work towards ensuring a future world rich in biodiversity. Thank you.
Thank you. And welcome, Team 2, Electron Technology to Maintain Genetic Diversity.
Good afternoon. Thank you, everyone. Next slide, please. My name is Pami Jasasan. Salmonella is a human foodborne pathogen. It is responsible for more than 1 million human foodborne illnesses in the United States every year. Next slide, please. Consuming or handling food, salmonella contaminated poultry food, meat and meat products are the primary source for human foodborne illnesses. current vaccines to control salmonella in poultry is not effective. Next slide, please. So I used electron beam technology, otherwise known as E-beam, to develop vaccines against salmonella. E-beam is a high energy beam of electrons that are generated using electricity. E-beam inactivates pathogens by damaging its DNA or RNA without damaging proteins that are essential to induce immune response. So next slide, please. So I exposed salmonella to a lethal E-beam dose and administered the E-beam inactivated salmonella as a vaccine to chickens. To test the vaccine effect of inactivated vaccine, I challenged vaccinated group of chicken and also a non-vaccinated group of chicken with live salmonella. Next slide, please. To test the vaccine efficacy, when we challenged with live vaccine, as you see here, we found a 99.9% reduction in colonization of salmonella in the vaccinated chicken. This proved that E-beam inactivated salmonella was a highly effective vaccine. I also use this for other poultry pathogens as well. Vaccine maintains healthy population. Healthy populations preserves genetic diversity and prevent economic loss. Thank you.
Okay, welcome Team 3, a recipe to fold genomics into the agro ecosystem.
Next slide, please. Hello, everyone. My name is Nathan Grant. I'm a postdoctoral research biologist with the Plant Genetics Research Unit in University of Missouri, Columbia. And here I am to talk about the ARSX Challenge Safeguarding Sources. We propose a framework to bridge genomics into the agro ecosystem to sustain genetic diversity. Next slide, please. The National Plant Germplasm System is a collaborative effort to safeguard the genetic diversity of agriculturally important plants. Conserving this genetic diversity ensures a robust pool of traits that can be used to develop crops resilient to our changing climate and that can overcome other agricultural challenges. The information and resources curated by the NPGS are hosted by the Germplasm Resources Information Network. GRIN is an open data ARS website that has aggregated trait data available for thousands of agronomic species. Next slide, please. For many taxa, next generation sequencing information exists for at least one reference genome and up to several thousand samples. However, this vast resource of genomic sequence data is frequently inaccessible for further characterization due to the lack of tools to manage, access, and connect the data to accessions or traits. We need a way to search genomic data for NPGS samples. Our solution is to create a framework that links GRIN accessions with applied genomics web-based tools. Next slide, please. We will build a framework to link GRIN with genomic data, starting with soybean, rice, and sorghum. Common bean is special in that it is just entering its genomics era and serves as a start-to-finish proof of concept taxa. GRIN users will be empowered to explore and select samples based on their custom genotype targets and list of variants or by alleles of any gene present in the organism. Curators can use this to prioritize accession characterization efforts by identifying sample duplications, gaps in genetic diversity, and help create more meaningful subsets within their collections. The interactive genomics search tools will increase the accessibility of upcycled big data. And a core idea of this framework is its modularity to apply in the future other agricultural organisms as they enter their genomics era. To safeguard the diversity in our germplasm collections, we need to leverage existing but inaccessible characterization information by folding it into an easy-to-use framework that links genomics big data with NPGS GRIN. Thank you.
Hey, welcome Team 4, Back to the Future, Old Genes for Modern Cows.
Hello and thank you. Next slide, please. Okay. Hi, my name is Caitlin Sarlo Davila, and today I'll be presenting on behalf of my team. Next slide, please. the problem. So modern dairy cattle have been through extensive genetic selection, largely for milk production. As a result, they produce more milk with fewer imports and are more efficient than ever. The downside is they have a weakened immune response and are less able to recognize pathogens. This is a huge problem for the US dairy industry, where the largest disease burden, mastitis, costs over $2 billion a year. And on top of this existing problem, we now see emerging diseases, such as the incidence of highly pathogenic avian influenza in dairy cattle recognized earlier this year. Since the week that I made these slides and submitted them, we are now up to actually 14 states and over 200 cases, making it more important than ever to safeguard our nation's supply of dairy. Next slide, please. So what's my solution? A unique herd in a living time capsule. At the National Animal Disease Center, I have a herd of Holsteins that has been unselected for anything since the 1960s. These cattle, in addition to being a unique genetic resource, are also mastitis resistant. So early studies have shown that when given the same initial dose of bacteria to cause mastitis, these animals are actually able to clear that bacteria and have less disease than our contemporary Holsteins. Germplasm from these is also stored at the ARS Animal Germplasm Program. Next slide, please. So my solution is bringing healthy back. So can we have our milk and drink it too? To basically create F1s between these 1964 lines and our contemporary Holsteins to preserve those immune advantages while also preserving milk production. Next slide, please. In addition to this and creating more resilient dairy cattle, we will also use a variety of cutting-edge sequencing technologies to be able to reveal parts of the genome, both coding and non-coding, that we've never been able to see before. Thank you.
Hey, welcome Team 5, the magic way to revolutionize peanut diversity.
Good afternoon. My name is Baozhi Guo from Tifton, Georgia, the capital of peanut research and production. Today, I'd like to talk to you and talk to peanut farmer about our magic peanut. Next slide, please. We all love peanut, right? Because the nutrition, the yummy taste. But the peanut also very special and unique in many, many ways, and face many challenges as I listed here. But the biggest challenge I want to talk to you is our farmer facing today. The production cost is increased, but the yield and the profit is flat and even decreased. Help on the way. Next slide, please. What have we done so far? We developed a magic population with 3,187 lines. Also, we used a subset of 310 lines of peanut, this magic population, to demonstrate the power of magic peanut. You can see the diversity increased, even we discovered new genes, new trait, and new alleles. Also, we built a pan genome. The pan genome powerful. It's very powerful because you can do the all-to-all gene accurate genotyping. Next slide, please. So here's the final pitch, our home run pitch with your help for our farmers, the winning pitch. We're going to build a pan magic database for better mapping, better genotyping, including one, sequence the whole entire population. Second, plus 10,000 unique genome sites. Number three, provide the unique, accurate genotyping for the entire population. Number four, we make the population available free to the community. Next slide. I'd like to introduce our team members from University of Georgia and Hudson Alpha. Thank you very much. Eat more peanuts.
Hey, welcome Team 6. What bee do I spy with my LiDAR eye?
Next slide, please. Thank you. Good afternoon, everyone. I am Mike Goldbush from Poplarville, Mississippi, and me and my collaborator, Sonia Dagan, who is part of Altoa Tech, have partnered to assist farmers, next slide, please, with assessing the need for pollinators in their cropping systems, whether they be honeybees, which are local or can be brought in from different locations, or native bees which nest locally in those farm sites. Next slide, please. Farmers have a diversity of bees available to them. Some of them, again, are native and nest near the crop sites, but honeybees are managed and can be relocated to places where there's pollination deficiency. Next slide, please. So to assist farmers in this challenging task, partnered with Altoa to use their technology to track insect flight behavior. And this tracking system allows for real-time continuous monitoring of bees at the site. It also is a non-destructive test. We don't have to kill bees to sample their detection in cropping systems. And then we can then use that information to also differentiate bees to species based on their flight activity. Next slide, please. And it's the impacts from this are economic. We can maximize the use of honeybees by placing them in places where there might not be native bees. We can also use the information to inform about ecological decisions. Can we use land management to increase nesting sites for native bees that nest near the farm sites? And then a global impact is then reaching out to monitor bees of concern, such as the rusty patch bumblebee. Next slide, please. So overall, we can deliver this information in real time. This can help growers, farmers make decisions quickly and to increase the amount of yield and production. Thank you.
Okay, welcome Team 7, diverse genes, robust crops, fortifying the future.
Hi, I'm Rachel Nagley, and I want to tell you about our plan for safeguarding our nation's food supply, diverse genes, robust crops. Next slide, please. $40 billion worth of food is lost every year from plant diseases, and that's a significant impact on our country. Next slide. And it continues to be so because management strategies take time to develop as pesticides can fail and new diseases emerge. But ultimately, this is an issue because crop genetic diversity is low, and we don't have a lot of information for many of the plants in the National Plant Germplasm System. Next slide. So the way the current system is set up is each plant is entered into the database. A user can then request and select those plants they're interested in. At first glance, this seems sufficient. However, MPGS has over 500,000 plants in it, and there's little information on disease resistance for many of them, which is one of the primary uses of this material. Our goal is to add disease resistance gene information specifically for each plant in the MPGS database. Users could then select the plants they're most interested in based on this information. This in itself would be really useful, but next slide, please. But we want to take those resistance genes. We want to predict their protein structure using AI, pair that with observational disease data, and then predict a role in disease resistance to fungi, bacteria, or viruses. Next slide, please. This would then be added back to the database as selection criteria, where a user interested in, say, bacteria resistance could then select just those plants that have that particular trait. This would be an amazing time and saving for money for both the user and the MPGS curators. But it would also fundamentally change how we're accessing and characterizing plant genetic diversity, as well as revolutionize how we're really thinking about and using resistance genes as a plant scientific community. Next slide, please. We're losing billions of dollars each year from food loss, but this doesn't have to be the reality for us. Diverse genes, robust crops is a vision for a future where food loss is no longer a concern and we have a safe and diverse food supply. I would also say that we like to think that this is a future where it's also easy enough to find and select resistance genes as, you know, buying your pair of shoes off Amazon, but we'll see if that actually ends up happening. But we have a world-class team with diverse expertise, and we're excited and ready, and we're hoping to make this the future. Thank you.
Okay. Welcome Team 8, safeguarding germplasm and admixture seed libraries.
Thank you, everyone. My name is Chris Stardick. I'm from the Appalachian Fruit Research Station in West Virginia, and it's my privilege to tell you briefly about our team's proposal, which focuses on perennial tree crops. So as many of you are aware, many of our tree species are under constant assault and threat from invasive pests and diseases, from climate change. And this just shows you many of the examples. And really, one of the challenges that we have as a society is how we help save these species and these industries that depend on them. Next slide, please. So currently, our system's designed where we have potential solutions in our germplasm repository. So these are collections of trees, wild species collected from around the world over many, many decades. And these have potentially crop-saving genetics within them, and lots of genetics that are just really interesting and exciting and will help improve our food. The problem for trees is because they take so long to flower from the time you plant a seed, that it takes breeders literally sometimes a lifetime if you're going to go back to a wild species and try to integrate a trait to help either save the crop or innovate in some way. So what our proposal is, the next slide, please, is to try to change this dynamic. And we developed this technology we call Percolate, and it really relies on two different technologies. So the first one is what we call pangenomics, and you've heard that mentioned already. The pangenome represents the genome of all the species within a genus. So for example, in apple, there are 35 wild apple varieties from around the world. So the pangenome would represent the genomes of all of those. And we can use that information to create kind of like a genetic catalog of all those species and their genes. The second technology is what we call rapid cycle breeding. And so this is a biotechnology-driven approach where we can shorten the juvenility period, or that flowering time in a tree species, from 5 to 10 years or longer down to just less than a year. And this allows us to do those crosses quickly and integrate those wild traits fast in a time frame that would allow us to potentially save the crop. So in this case, what we're proposing is to really create what we call like seed libraries. Next slide, please. Where what we would do is integrate the wild genetics and preserve them within domesticated crop backgrounds. So we're not just providing wild tree species, we're actually providing breeders and growers with seeds that are already mostly domesticated, but have just the small pieces of genetics that they're actually looking for. And so that shortens the time it takes to develop a new variety to just maybe one generation away. So that'll stop. Thank you.
Now, Team 9, crowdsource characterization for genetic resources.
I'm Sarah Doley from the National Plant Germplasm System in Washington State in Pullman. So the National Plant Germplasm System, next slide. The National Plant Germplasm System consists of 22 gene banks across the US, which maintain collections of over 200 crops and their relatives. We freely distribute over 200,000 samples a year, primarily in the form of seed packets, which researchers and breeders use to create crop varieties to meet evolving human needs. Next slide. For example, the bean collection, which I manage, contains over 17,000 samples and distributes around 5,000 samples a year. However, we only have capacity to grow seeds for 300 samples a year. We lack staff and field space to grow and characterize our collection. Next slide. SeedLinked, founded by our team member Nico Engelbert, is a startup company which has cultivated a network of over 10,000 volunteer growers who run plant variety trials around the world. SeedLink has developed a user-friendly smartphone application for trait data collection with a social network component so growers can answer each other's questions and build community. This map shows the current US-based volunteer network. This project will bring together the GeneBank's diverse seed collections, including beans, ornamentals, and vegetables, and the SeedLink growers to generate multi-environment trait data at a fraction of the cost of traditional research trials. Next slide. As proof of concept, we're running a bean pilot project this summer. The project recruited 200 volunteers in about an hour to trial 20 bean lines, demonstrating the huge public interest to participate in germplasm characterization. Next slide. With ARSX funding, we will generate characterization information for over 150 germplasm samples across crops utilizing 1,000 volunteer growers in the first year, and create an updated version of the SeedLink platform to streamline integration with the gene bank data collection and management system. In the long run, this platform could be used by all gene banks as a cost-effective method to generate multi-location plant trait data. So join our initiative to phenotype all USDA germplasm, and this spring sign up to be a seedling volunteer and characterize part of our collection.
Thank you. Thank you. Can we have a round of applause for all of our teams after those presentations? I am sure I could not do that. I'm positive. So now I'd like to invite Dr. Kappas up to introduce our keynote speaker. Welcome, Dr. Kappas. Haven't seen you here in a bit.
Thank you, Dr. Cook. And it is great to be back at Beltsville and see many of my friends. I have the honor this afternoon to introduce our keynote speaker, Dina Lampert. Dina is the Inclusive Innovation Lead for NASA's Space Technology Mission Directorate, located at the NASA headquarters here in Washington, D.C. This directorate's core mission is to enable aerospace missions through research, development, demonstration, and testing across multiple sectors. She is originally from Little Rock, Arkansas, and she currently resides in Greenbelt, just around the corner from here. And she resides there with her 5-year-old son, Caleb, and her retired guide dog, Angie. Her work in the Science Technology Mission Directorate is to advance research opportunities for innovators in underrepresented communities, including small businesses, new entrants, and adjacent tech sectors. She supports the development of NASA-funded research opportunities, outreach efforts, and resources for those seeking to engage in NASA's technology pipeline. In 2022, Dina was selected as the Astro Access Ambassador and completed her first microgravity research flight in December of 2022 and continues to research efforts for accessible space flight. With that, please welcome Dina Lampert.
All right. Thank you so much for having me here. It's nice to get out of the aerospace bubble and actually learn a couple of few things. I've heard about bees and seeds, so this is definitely exciting to be here. All right, let's see. Do I have my slides up? Okay, next slide. So one of the things I was talking with a friend of mine, and I was letting her know that I was coming here today, and she's like, they do this type of work. So I would say in Greenbelt, you have the enthusiastic support of a mom of 2 children who are excited that you are doing work in gene diversity. Next slide. As mentioned, our work in STMD, Space Technology Mission Directorate, is to ensure that the research pipeline for NASA is ready and able to supply the technological needs of our missions in the future. So I hope for that Charlie's Angel picture up there that I can show that definitely to my 5-year-old son about 10 years from now that yes, I did have a cool job that allowed me to go and do things that I otherwise would not have been able to do, or that I'm not cool enough to do in the future. So next slide. All right. So I know a lot of folks may think of NASA as being the component that has manned missions or a rover on Mars, but there's a lot of capabilities that you see within NASA that enables not only our discovery in space, but also terrestrially here on planet. And I think that has similar applications to the work that you do here and across the country, in that we see the name or label USDA and we think of our food at the grocery store and that's a seal of approval, but there's a lot of things that work behind the scenes that makes it available so that we do have a healthy pipeline of food and plant resources. So for us, we have a functional capability that lines up well for GO, which is our basic space transportation. Land is once we're able to land there and have surface transportation. Live is once we're able to land, we're able to use the infrastructure on the surface and explore that surface area. Expand is in-space infrastructure, and enable is foundational capabilities. In these functional areas, if you see kind of the imagery from the Earth, Moon, and Mars or beyond, we utilize the expertise of the aerospace community, both within NASA and within the ecosystem, to say, hey, if we want to have these missions 10, 15, 20 years down the line, what are the technological capabilities that we need to invest in now that ensure success in the future? So next slide. So some of the specific things that you may have seen on the news is this past year, we initiated and had our first mission of deep space communication under Psyche. So there was our first transmission from more than 2 million miles away. And some of you who may keep an eye out for CATS, it was our first video transmission from 2 million miles away of a cat named Taters. So cats have been winning this year. The next thing was from our radio frequency mass gauge. And that was essentially from our CLIPS mission, which we are creating kind of our prime delivery on the moon, where we utilize our commercial partners for that type of transportation to the moon. But where we're able to get from that mission from Intuitive Machines was valuable propellant data. And then finally, we launched our Starling swarm capability for multi-CubeSat function. And that again is utilizing commercial partners from SpaceX and was a demonstration project that is still ongoing. And then finally, we're using fission power for nuclear demonstration. And again, that's an industry-derived concept and is currently ongoing through 2024. All right, next slide. So what you are seeing here within the ARSX competition here is a function that a lot of federal agencies are just stepping into this capability under what we call the Competes Act Authority, but it's essentially our prizes, challenges, and crowdsourcing. It is where it's saying for the government, whether we're talking NASA or we're talking USDA, that we're saying that, hey, we want to generate an incubator of ideas, whether it is from our workforce or from externally. We want to have the capability to provide incentives for those disruptive ideas. I do not like getting a phone call from my son's teacher saying that he has disruptive ideas, but we do like to have those disruptive ideas encouraged, nurtured, and ultimately supported. So for instance, we have a small orbital debris challenge that originated at as a NASA at Work challenge from our employees that then culminated with an external challenge for remediating debris in low Earth orbit. And then finally, we have an example of break the ice challenge. If we're able to go to the moon, we have to be able to break below the surface. And what we're hoping is that there will be technologies that will be spun out that will provide efficient, cost-effective robotic access for terrain excavation and construction. All right, next slide. So one of the things that we we took a look at a couple of years ago and said, we need to encourage a diversity of ideas. If you look at our proposer pipeline, we kind of sometimes see the same proposers over and over again. And that becomes very homogeneous in the types of methodologies that you see coming in. And so we took an effort to say, hey, what are the barriers to entry for people coming in? And if you all go to conferences, you're talking with your farmers and out there, and some of them say, hey, we didn't know that either NASA did this or USA did this, or would be concerned about these types of concerns that we have. So we hear people say, hey, I didn't know about it, or I did not know how to navigate or provide input into this process. Or then in a competitive sense, when we do look at those opportunities for engaging industry and academia, we don't know how to respond because there's too much of a barrier for entry, whether it is providing too much of a complicated process or something that prevents them from engaging. So it essentially falls into categories of saying, what do we say? How do we move? Where do we show up? Who joins us? And how do we provide access for them to come in? And then ultimately, how do we grow that ecosystem? So for those that are here from North Dakota or from Missouri, how are you inviting the community around you to see the work that you're doing and be excited and ultimately support the work that you're doing? All right, next slide. So this kind of helps us fall into three categories of encouraging diverse perspective, challenging the norms, and then creating a space for collaboration. Next slide. All right. One of the things that what we've seen that you may have implemented here in ARSX, or at least in competitions, is really looking at how we can make our streamlined approaches. Sometimes doing a 50-page proposal, why do we need that if an idea can be articulated in, let's say, a pitch competition or in a two-step process or in open submission, rolling submission deadline? The other thing is, how can we create a community where we meet people where they are? We look at the challenges that they have that are most relevant for them. How can we rely on the expertise that we can bring from our workforce, which is what you're doing here? And how can we create communities of mutual benefit and exchange? Next slide. So this is where it's taken kind of us being able to go outside of DC and where we've been able to go. One of my projects that I've done this past year is our Space Tech Catalyst Prize. What we know is that regionally there are some ideas that are cultivated at the local university or lab level that we really would like to take notice of at a federal agency level, but we need to work with the intermediaries or mentors out there. So we did an effort to identify those individuals and organizations. We're hopefully doing some community building and capacity building with them. And then what our hope is that they can go back and then spread the word on our behalf as trusted messengers within those communities, help us redefine or adapt our process so that we can capture as many of the solutions and then in the future see them in our pipeline. All right, next slide. So what does this translate into? So a couple of the competitions that we've run inside of NASA is our strain measurement for Kevlar and for habitat structures. One of the things that you notice, if we are going to land on the surface, we're going to use in situ resource access, we need to have habitats that are able to withstand kind of the flexion or deflection in space. So one of our employees said, hey, I'd like to bring this challenge out to not only our workforce, but externally as well. And one of the things that with a little bit of money, I believe they had $40,000, he was able to come up with some approaches that was so simple, so elegant, and how could we have not thought of this? And that is speaking to the benefit of diverse ideas coming in and where we're able to hear and receive those. Next slide. All of you have come across some type of screening process through TSA. One of the things that they needed to do was to have a multi-millimeter wave algorithm that would allow them to detect threats earlier with a lot more precisions. Again, they went through, they identified 8 winning algorithms that were developed, some at about a 98% detection rate, which was really good for them. Now, at this level, you're looking into the multi-million-dollar projects, but for them, it was well worth it and valuable for them. And again, these are things that are either used as crowdsourced at a lower level that people were able to contribute their ideas. Next slide. And then finally, one thing that may be kind of closer to home is the astronaut dietary food intake tracker. I believe one of the ideas that I heard here was, can we use a software function to solve or at least be able to understand how much our astronauts are taking in, that is intuitive, that provides that user experience that is simple, but gives us enough information that is reliable and we can duplicate over time. So again here, what we are able to is to take a systems approach and break that requirement up into smaller components and utilize the expertise of multiple developers that were able to come in, contribute their component, and ultimately come up with an app off of the iOS system that is currently being used on the station now. And then finally, what I would say is going back to, next slide. All right, so going back to kind of the problem that we started out with is we often find that we have these known technology challenges either within NASA or here in ARS that you need to solve. And what I really love seeing with you all that is you're using the knowledge, the curiosity, the questions that are being asked, and the resources that you're putting towards answering those questions and bringing in diverse perspectives so that we can shift from people not knowing, or at least the community of researchers not knowing, how do we participate or how do we bring these ideas forward? I've been working in my lab. I'd like for someone to be able to notice these things and ultimately grow the readiness level so that we can either spin it out towards commercialization or bring it as a standard. But then we also know that we need to invite the community in to learn to say, hey, this is how you move and navigate within the competitive process. And then ultimately, we can shift from awareness to active engagement and applications so that we can see folks and you all here as 9 teams saying, hey, I want to try, I want to present this. And so that's where I would go back to the Wayne Gretzky quote earlier of 100% of the shots taken are not known. I believe that's how it goes. What I'm glad is that there are at least 25 teams that took your shot, brought it here, and I would say good luck and congratulations.
Thank you so much, Dr. Lambert, for that inspirational, informational, and informative talk about disruptive science across all our communities. And I think that's what we're trying to achieve here for sure. Okay, I want to invite Dr. Simon Liu back to the podium for our final presentations. Dr. Liu?
Well, what a presentation. I really enjoy, even though that is the second time I hear the presentation with a shorter version, but still so impressive. Thank you again for all of you here. Thank you. And also, I'd like to thank our friend from NASA, Ms. Lambert. That is a really intriguing presentation here, very, very interesting. This reminds me of my good old day. I worked for NASA Goddard Space Flight Center here in Greenville for 10 years, primarily working on the space station project and our Earth Observing System project and a Landsat project. So 10 years working only on 3 different projects. This reminds me of the good old day. Thank you. All the innovation, all the challenge we have, not only on the ground, but also in the space. Thank you. Thank you for Ms. Lambert for your presentation. Thank you. So we come to the stage. I'm sure that you all anxious to it. You're very anxious to it in terms of who are the owner. But wait, before we get into that, we need to do 2 things. Good. So the good thing is always worth to wait. Okay, the first thing is let me invite all the team members come up here for a picture. So yeah, team members coming up. Is it individual team or a team member? Okay, come on here, and then we have a picture. Right, we need to kind of concentrate a little bit closer so I come by. So, all right, thank you all.
Thank you, that works.
One more thing I need to do before we announce the winner of the competition. I can kind of show you back in the stage how the processes work here. As I mentioned earlier, we have 25 entries, 25 application package. So the ARS team led by Dr. Silverstein and also Dr. Penn Stockery and others, they do the first initial screening. The screening based on a few criteria. Of course, the first criteria is innovation, how innovative the proposal is. After all, this is the ARSX. So innovation is the main theme, right? So first, the innovation. Second, what are the potential impact? Because we say this is a high risk, high reward competition. So what are the potential reward? What will be the potential impact? So those are the first 2 major criteria there. Now, the third one is we are looking at your research hypothesis, your assumptions, or maybe your methodology behind the scene to ensure that we have the good approach, the physical approach, and to see if your approach is also innovative. So it's really assumption, hypothesis, and your approach. Just like we do our dissertation research. You not only to look at all the state of the art, but also you need to look at the methodology. The fourth thing we're looking at is your team composition. Do we have a team that has the right skill? Do we have the team has the interdisciplinary skill necessary to not only implement the project, but help to imagine the possibility. So the team composition is very, very important. And also, are you only working from a lab from your own? Or you working with the team members across different laboratories, across different disciplines? So those are four major criteria we use. And the last one is that, as you know, that each judge, they have their personal preference, personal endeavor, personal interest. So if your proposal really fit into their personal interest, personal endeavor, and that will be a few extra points will be added to it. So those are the 5 major criteria we use. The same criteria used at the first screening process by the ARSX team. And then after that, we got 20 projects, reduced from 25 to 20. So the 20 projects submitted to the administrator's office. So 3 administrators, one of them is sitting there, the acting associate administrator, Dr. Martin Eve and June Park, and myself. We look at those 20 proposals and begin to evaluate based on the same 5 criteria I just talked about. Okay, we each score it, and then we get together to have a meeting. So 3 of us are sitting together after burning the midnight oil during the weekend, because ARSX team is very effective. They always give us only a weekend. Right. So we spend the time in the weekend reviewing all the proposal, and then Monday we get together and begin to select 10 out of 20, which we did. Of course, there are some debates. There are some looking at from different criteria perspective, the debate, and then finally we come to the conclusion, select 10 out of 20. So the final 10. Get into today's competition for all of you here to present to yet another panel that are the judges. I'm the only one maybe go through the second round, the third round. So the rest of 4 panelists, they all brand new, they're from outside. So that we will be able to looking at it from the inside perspective, but also the outside perspective. So after your great presentation. So right now, we come to the point that I will announce the winner. Where is my sheet of paper? All right. So anybody try to drum it? All right. All right, the very first one, again, is not in priority order. All 3 are awarded with the grant. First one is diverse gene robust crops, fortifying the future. So the winner, please stand up. All right, okay, all right. Hey, please stand up. You have.
Thank you.
Right here?
Oh, one more. Okay, sorry. Pausing.
Or just stand together without. Okay, here we go. All right, thank you. By the way, I like your answer to my question earlier in the presentation. I asked the question, say, how can your project increase the diversity? And your answer is that, well, we're not increase diversity, not only from the germplasm perspective, but also functional diversity perspective, which is good. And your answer about the level of effort required. So you say, well, this project will be able to help us. to focus on more high value, high impact germplasm. Thank you for your insight on that. Thank you. All right, the second team. The second team is safeguarding germplasm at mixture seed library. I like your approach. Remember in your presentation, you say one of the major characteristics of your proposal is you will be able to re-engineer so the crop, especially woody crop, they will be able to flower faster. Instead of multi-year effort, you can cut it short into 1 year flowering. Teach me how, because I have a tree in my backyard, never flower. All right, thank you, congratulations. Now, the third winner is the crowdsource characterization for genetic resources. Congratulations. I must say this, ARS, as you know, that we have 22 gene banks at 22 locations out of our 95 locations around the country. 22 of them. Some are big, some are small. So to ensure that we preserve those genetic resources is a major, major challenge for ARS as an agency. As you know that to do so, we need to first collecting our germplasm, regenerating our germplasm. Not only that, but also we need to store those germplasm. We need to manage those germplasm. We need to characterize those germplasm. We need to preserve those germplasm. We need to disseminate the germplasm to our stakeholders. A lot of work needs to be done. So as a result, a huge amount of resources we have really invest in this endeavor. The germplasm for ARS is a national treasure. We need to make sure that we continue to maintain, not only maintain, but also continue to grow our national treasure. So in order to do so, we need to have a good, innovative approach. And this is what this competition is about. So I'm glad that you're outsourcing here. We have been thinking strategically how ARS we can maintain those 22 gene banks. How? Can we do more, faster, cheaper, and provide better service to our customers? That is the thinking. Shall we centralize, make it bigger, or shall we decentralize? Or shall we maintain the current 22 gene bank? This is all really a part of the discussion. OMP, the national program leader, we are thinking through those process, different scenario with one goal. To preserve, not only to kind of preserve our genetic resources, to help our people, our stakeholder, so that we can enhance our ag production system, enhance our ag protection capability, but also preserve the nature of those germplasm for future generation to come. Now, in the meantime, I'm going to issue you a challenge. Our NASA friend is here. You know, growing anything or raising any animal, you need to have a few major ingredients. You need to have air. You need to have water. You need to have soil. In a space, no air, no water, no soil. So I challenge all of you here, think behind the scene. How do we grow the food in the space? for our future endeavor. Our future endeavor. I'm sure in the future, people will travel to the space, not just travel there, maybe stay there. How do we provide the food for them? Think about it. Maybe in the future, this become a major challenge of us. ARS, we do have some work along those lines, working with our friend in NASA through the interagency cooperate agreement. But I'd like to challenge all of you here, thinking of growing food in a very extreme environment. Of course, climate change is going to impact that. All right, so with that, for the rest of the team, 6 teams, even though you did not win the competition, but again, you all are the winner. Every one of you. You should be proud of you to make this point. And you should be proud of you to be able to present your idea in only 5 minutes. With a combination of the PowerPoint and your spoke word. It's not easy. It's a challenge for each and every one of us. How do we capture the idea, the concept into a form, be able to present to the people in such a short time frame? Not easy. Congratulations to all of you here. This is also a part of our ARS project, the goal. The goal is that provide a training opportunity so that our scientists will be able to go through this process to equip themselves in terms of how best to capture the idea, how best to present the idea, how best to convince people to invest in those ideas. You have done so. Even though you don't have it today, get an award today, but we still have plenty of other opportunity. I encourage all of you here to talk to your national program leader. Of course, participate in the Innovation Fund competition, participate in the postdoc competitions. And every year we have hundreds of postdocs across the agency. There are plenty of opportunity, and talk to your national program leader. Your great idea, I don't want to waste it. I like to fold those great ideas into our research program, into our research agenda. Even though our national program, they have the so-called program plan, but they are flexible. If you have a great idea, we need any time so that we can continue to infuse those innovations into our research endeavor. And with the goal to speed up our discovery, to serve our stakeholder, to provide a scientific solution that will be address the challenges that face our stakeholder every day from field to table. That is our mission. Thank you all.
Thank you, Dr. Liu. Thanks for those comments. And thanks to our judges, everybody here and online, and especially thank you to our finalists for being here. Please, let's give them another round of applause. Okay, have a great afternoon, everyone. Bye.